Okay, can everybody see my screen?
Yes.
Okay, great. So again, just a quick reminder, my name is Andrew Blair, and I am a graduate student at UCSF. My family has a history of congenital heart disease, and so there was an opportunity to design a massively parallel reporter assay with a consortium called the Impact of Genomic Variation on Function in the AHI-2 lab. And so today I'll be reviewing, again, some of the leading questions for the design of this test cardiac MPRA investigating congenital heart disease, as well as some of the open source applications that we're developing to help uniformly process these data sets. All right, so for a disease background, 60% of CHD cases have an unknown cause. Nearly 90% of all phenotype-associated single nucleotide polymorphisms identified by GWAS lied within non-coding regions. And so genome-wide association have implicated common non-coding variants in congenital heart disease, for example, for atrial septal defects and left ventricular outflow tract obstructive lesions. And so to investigate these non-coding variants, the study data set generated by the Pediatric Cardiac Genomics Consortium, or PCGC for short, identified patients, or a trio proband with patients with structural CHD and their parents. So there were 763 trios that were enrolled in this program. And the controls were comprised of about 1,600 autism unaffected sibling parent trios, and this is from an autism quartet study. And so this analysis was one of the first research articles that I reviewed in Benoit Bruneau's lab when I was a computational research associate. And it was just a really inspiring research article in terms of all of the work that went into identifying de novo variants and then integrating in multi-omic assays like H3K27 acetylation here for prioritizing enhancers, as well as building these neural networks for annotating these regions, as well as measuring the effect of the variant on transcription. So this was, again, kind of the leading article that inspired me to come back and do my PhD and investigate congenital heart disease. But there are some limitations to deciphering non-coding variant mechanisms in CHD. And one is just a lack of understanding the molecular consequences of non-coding variants. And there's also limited annotation in the delineation of these regulatory elements. And then finally, these non-coding variant effects might be cell type specific and are associated with distinct developmental time points or transient cell states. And so this, given these limitations, it leads to this question: well, how do we measure the level of activity for non-coding regulatory sequences and variants that are associated with congenital heart disease, given that regulatory sequence sites are important for transcription? And when we look at this little cartoon diagram here, we can see that we have a promoter region interacting with an enhancer. And we have this remodeling complex guided by transcription factors that are helping facilitate the expression of this gene here. And so when there are SNPs or variants within these promoter and enhancer regions, we want to be able to evaluate how these variants impact the expression of these genes. And so the goal for my research is to investigate if there is an enrichment in regulatory element categories among CHD de novo variants. And the functional assay that we'll be discussing today and employing for investigating this question is called massively parallel reporter assay. And so what is a massively parallel reporter assay? Well, it's an assay that can functionally validate thousands of regulatory elements simultaneously using high throughput sequencing and barcode technology. So it starts first by identifying candidate regulatory sequences. And so these are traditionally identified using some chromatin state model, like a hit Markov model, for instance, or guided by other omic modalities like ATAC-seq and ChIP-seq, and looking at different combinatorics of accessible chromatin and histone ChIP-seq marks. And then also we have a minimal promoter that drives the expression of a reporter gene, so in this case, luciferase. And then finally, we have our candidate regulatory sequence that has a unique molecular identifier so that we can identify the transcription of these CRSs and link them to, again, a barcode. Once we design and select our candidate regulatory sequences, we need to have a delivery method. And so there are a couple different methods for that. There's lenti MPRA. This is a experimental technique that Nadav's lab pioneers. And then there's also adenovirus. And within this, there are different transfection modes of integration. So episomal versus chromosomal. And then once you can get those experimental parameters defined, you do a RNA DNA extraction from the cell sequence, and then you quantify the activity score. So this is just a ratio of the RNA and DNA. And so the question that started when designing this first assay, and I'll open this question just kind of broadly to the community right now, is what functional genomic assays and methods/models would you use to select candidate regulatory sequences that harbor CHD variants? So I'll give it a couple seconds for people to think about it, and feel free to post it in the chat or raise your hand or go ahead and turn off your mic. Okay, going once, going twice. Okay, let's keep going. So to identify different types of genomic states or chromatin states, typically we look at different omics modalities. So for instance, identifying chromatin accessibility, we employ an assay called the TAC-seq. And we can see that when we look at chromatin accessibility with other omics modalities like DNA methylation or these histone marks, we can see that there are different distributions within the cell. Specifically, we see, like, for instance, bimodal distribution along the chromatin accessibility mark to identify active promoters, and similarly with enhancer poised or primed regions. And so we use a combination of ATAC and ChIP-seq and RNA-seq to select these regulatory elements that we want to measure the activity of the sequence itself using MPRA. And so when we want to think about how do we then design this assay such that we can ask questions where we can see, for instance, an enrichment of CHD variants within a specific regulatory annotation, like active promoters or poised/prime enhancers, we want to be able to have some background guiding library data set so that we can develop a testable hypothesis to say that, for instance, if a CHD variant is harbored within active promoter or an enhancer region, that we are able to say that it has an activity score greater than some background control set, whether that's a positive or negative control set. We want to be able to design that testable hypothesis. So that leads us to thinking about an enrichment analysis of cardiac regulatory elements. And so we want to be able to associate cardiac gene sets or pathways with a background set. And once we define these background sets, then we can start to develop a null hypothesis and test statistic, whether that's a bootstrap or permutation test. And some of the software that I use for that is RegionR, Annotator, and Annotation Hub. Although Mike Love's lab has null ranges and bootstraps, which is a much more heavy-duty version of these software tools. So anyway, so for example, one enrichment analysis might be, is the cardiac ventricular development pathway significantly associated with congenital heart disease de novo variants? And so first, you'd want to do a background screen of this GO pathway. So there are 536 regions that are associated with this pathway, and that's intersecting with your background set. So now we'll want to define a null hypothesis to evaluate. So in this case, our null is do gene ontology, this ventricular septal development associated region, and CHD de novo variants overlap the same amount as what would be expected between our de novo variants and random regions. And so we depending on your null hypothesis, you can develop a test statistic, in this case, a permutation test to evaluate whether there is a significant association between this gene set pathway and your de novo variant gene list. But again, you still need to have some background data set to be able to select and identify where these regions are in your experimental model. And so to define that background data set, I use the Cardiovascular Development Consortium, or the CVDC version 1 data for selecting candidate regulatory elements. And so quick background on this data set, we had an experiment. So this is an experimental in vitro data set in H9 iPSC cells. And there's a differentiation between day 0 and day 80, where at each time point, we had 2 biological replicates per time point in assay. So again, day 0, we have all 4 of these genomic assay modalities and 2 biological replicates. And we were using the human embryonic stem cell set, which comprised roughly 96 data sets, to develop this testable hypothesis where we're wanting to really just define or test region background set so that we can start to develop a null hypothesis evaluating if there's an enrichment of CHD variants in regions that are associated with a specific developmental process or disease process in cardiovascular development. So this is a high overview, a look at our MPRA test design for investigating the activity of sequences and variants associated with CHD. So to begin, I did a literature scrub on congenital heart disease and cardiometabolic associated genes. And I used our CVDC version 1 background data set to identify these genes that have that are expressed within the RNA-seq. And then I used the Hi-C data set to identify their topologically associated domains. So to do that, I took the lower bound of the start of the TAD boundary and the upper bound of the end of the TAD. So that defined the regulatory domain for each of our target genes. And then we developed a selection ranking strategy, or in this case, chromatin states. So we looked at different combinatorics of attack in 27-acetylation and H3K4 trimethylation to identify, for instance, candidate promoters and candidate enhancers. So, for instance, we have this CHD/cardiometabolic associated gene, and we're selecting all candidate promoters and enhancers that are within some linear distance of that target gene and intersect with other targets of interest. And what I mean by that are just some a priori background data set that intersects with these regions. So for instance, the CHD database, or the IDACUR and Chi Labs CHD and Autism Convergent Molecular Network. So we intersected with also the Vista Heart enhancer. So lots of a priori information went into intersecting these candidate promoters and enhancers. And after we went through the selection ranking, anything that was left over, we just included within the library. And to evaluate if any of the Hi-C contacts, so for instance, here we see a candidate promoter and a candidate enhancer that are in contact, identified again by the Hi-C. And here we see that the enhancer and the promoter are in contact, and this promoter is also in contact with the protein coding region of our target gene of interest. We want to be able to backtrace and evaluate if any of these Hi-C contacts are indicative of differential activity within our MPRA assay. So, and this is important for temporal modeling, but in this case, we focus just on a small window, primarily the cardiac mesoderm cells, cardiac progenitors, and primitive cardiomyocytes. And the reason for this was we want to be able to define test regions that have high activity in progenitor and primitive cardiomyocytes that can be used in much larger MPRA screens that are evaluating an allelic series of CHD. So when we're thinking about designing an allelic series or an MPRA evaluating an allelic series for CHD, we want to consider how we're designing our oligos for an MPRA. And so for this test MPRA, which again is just to define regions that have high activity at the progenitor and primitive cardiomyocyte stage, we wanted to be able to ask where is, when we center the oligo to test in the MPRA, does the placement of the oligo have a different activity level when we're centered on the variant, or in this case, the summit? So what is the summit? That's just where the number of reads pile up at either an accessible chromatin region from ATAC or a histone ChIP-seq mark. So we've got, in this case, for our enhancer region, we just centered at the ATAC-seq summit. And for our promoter regions, we had two different tiling strategies. And there were some experimental limitations to this design. So for instance, we're only able to evaluate 270 base pair. And so for promoter regions where the summit was closer to the start of where the regulatory element began, we just did standard tiling. So we started where we first saw the signal, and then we started to tile over with a 90 base pair overlap. And the idea for that is that if there's any transcription factor binding sites, we wouldn't be missing that with that 90 base pair overlap. But if we saw that the summit was in the middle, and we had some conditionals here where if the location is greater than or equal to 350 base pairs from the start or end of the regulatory element, then we would just center there and then tile outwards. And so again, the idea here is that we want to be able to measure, when we do develop the allelic series, where do we place the oligo with respect to the variant? Do we center the oligo on where the variant is at, or do we do it closer to where the variant is at the beginning of the oligo? And so in this preliminary test MPRA, we're wanting to develop some in silico models so that we can identify where we are placing those tiles. And that's in collaboration with, again, the IGVF consortium with Anshul Kandaji's lab. So, I've talked a little bit about positive controls, but I haven't mentioned negative controls yet. So when we are evaluating if there is a differential activity in an allelic series, we want to be able to say that, for instance, the alternative sequence has lower or greater activity than the reference. Here we're wanting to define our negative control background set. We want to be able to select regions that and design them in a way where they should have no regulatory activity or no activity score in the MPRA assay. And so for these negative controls, we randomly selected 50 sequences from each assay type, and then and that included the different combinations and overlaps. Then we randomly shuffled. We did a dinucleotide shuffling on each of those random sequences that we pulled. And then for the other negative control set, I'm calling this the candidate negative control set. These were regions that were randomly selected from the TAG boundaries. And when we looked at this within the UCSC genome browser, we saw that there was like no signal also within our guiding library data set. And so here we're also evaluating if any of the high-C inference models that define the boundaries and the contact regions have what we're expecting experimentally. So, and again, in summary with that, we want to be able to evaluate if the high-C inference models indicate a differential activity based on the contact scores between promoter and enhancer regions in the target gene. And then the second point for this is to evaluate if the boundaries that are identified by high-C are really delineating regulatory domains. Okay, so in summary, we selected 652 CHD and cardiometabolic associated CRE targets and within their TADs. We use the high C to identify a lower bound for the start and an upper bound for the end. And we did some thresholding for the RNA-seq. So we made sure that, given our literature scrub, that we did see RNA-seq signals within our guiding library data set. And then we prioritized our candidate regulatory elements using combinatorics of attack and ChIP-seq, as well as any intersections of our target of interest. And for our negative controls, we took, again, all of the different combinatorics of our attack and ChIP-seq and did dinucleotide shuffling. So that's just a standard procedure for designing our negative control set. And then for our candidate negative control set, we randomly selected sequences from our high C identified TAD boundaries. So the idea here is that attack and ChIP-seq, these are low resolution assays that just kind of give you an identification of where in the genome you see accessible chromatin and these histone marks, but you're not getting like a quantitative signal of the sequence itself. And so here, what I'd love to build up to is to start to think about building a multi-omic molecular network where we're able to define or layer over the activity scores of the sequences identified from MPRA and use MPRA to guide a CRISPR-I screen. And so I like to think about all of this in the context of a network. So here is just an overall design of a proposal to construct a multi-omic enrichment network. So you go in the genome browser, you see all of the tracks. You got your enhancers, promoters, your target genes. I like to think about collapsing all of this within a network. And here are the color gradients just correspond to enhancer, promoter, and then our protein coding region. So the idea is that attack and ChIP-seq, this can identify our enhancers and promoters, but it doesn't give you like an empirical signal of the activity of the sequences. And so that's where the MPRA assay comes in. And so the color shading within the network here represents the activity level of these sequences. And when we intersect these sequences with whole genome sequencing, for instance, we can start to be able to develop these enrichment strategies where we're evaluating if an enrichment of a CHD phenotype within a promoter region has an impact on transcription, and then identify at what point in time does that variant impact the developing heart. And so for instance, you have an enhancer that has high activity at the mesodermal cardiac stage. And we see here with these links here identified by the high C that the enhancer has strong contact with the promoter. And this promoter region harbors a CHD variant. But at the mesoderm stage, we're not seeing high RNA-seq signals. And so when we go down the differentiation into the progenitor cardiomyocyte, we see some differential activity in the enhancer region and the promoter. So enhancer activity start to go down, promoter goes up, as well as our RNA-seq. And we're also starting to see some differences within the high C contact. And at some point we see that the enhancer is no longer active, but the promoter is active and the RNA-seq levels are high. And so we might be able to then devise a null hypothesis such that we're evaluating if at the progenitor cardiomyocyte stage, the CHD promoter is impacting the differentiation of progenitor cardiomyocytes to primitive cardiomyocytes. And we can start to layer on, tertiary experiments evaluating using CRISPR-I or maybe CRISPR-A, where if we silence this promoter, is it also silencing the transcription of our target gene, and is that having an impact on differentiation from progenitor to primitive cardiomyocytes? So given the lead up to this multi-omic enrichment network, we also want to have some uniform processing of our MPRA data. So the biodata catalyst already has all of the ENCODE, attack, and ChIP-seq and whole genome sequencing workflows, as well as RNA-seq, but we don't have a massively parallel reporter assay workflow for processing each stage. And so Over the last couple of months, Dave has been absolutely phenomenal, helping train me to get the MPRA workflows up on the biodata catalyst, as well as some of the ranking models to select these regions. So our primary computational wheelhouse for MPRA starts with MPRA snakeflow. And so what this does is it associates barcodes with sequences. So that's the first part of of your experiment, is looking to see if your barcodes are promiscuous with other candidate regulatory sequences. And then we want to count these barcodes for the DNA and RNA. And then we have another tool called MPRA Analyze. And what this does is it takes our sequence pairs into our reference and alternate alleles and does a differential expression of these sequence pair reads. So, you know, in the biodata catalyst, what we're doing is we're building a push button application for processing workflow that we'll make as an open source application on the BDC. And so what we see here is just an info description panel of what this looks like. So we have some tags here for hopefully people that are searching for the workflow, they'll be able to identify it with some of these key terms that we float around in some of the other consortium work that is supporting this these studies. And so we just have an overview of how the workflow is broken down into these two different CWL tools. And these are just a stitching, the assignment and experiment step, or a stitching of MPRA snakeflow. And I think Dave might talk a little bit about this, as well as some of the ranking models at the end. So, you know, we have our processing, and then we have our analysis software. And what I've been primarily developing over the last couple of months is an MPRA database. And the idea here is that we'll have uniformly processed MPRA data sets with minimal standard operating procedure for our primary, secondary, and tertiary analysis. And you know, the background for this is that there have already been some prior cardiovascular research studies utilizing MPRAs, and to date, only 2 cardiac studies have been published using MPRAs, both from Bill Poo's lab. The first work was done identifying genomic regions that were bound by multiple core cardiac transcription factors. And the library here was a little bit different. So it was a library that evaluated 2,700 regions. And in this case, the regulatory elements that they were able to evaluate were 400 base pairs in length. This was also an in vivo assay. They had a different way of defining their background positive and negative control set. And then somewhat recently, they had a second cardiac study that expanded on the results of how they selected regulatory elements in this Nature Communications article. And here they were identifying candidate enhancers that included a subset with atrial or ventricular specific occupancy. So I'm going to show just a quick teaser of what the database looks like right now. What you're seeing is the landing page of the database. Can everyone see this? Do I need to change?
Yes, we can see it.
Okay, thank you. So what I'm showing here is just the landing page of what the MPRA database looks like. Here, each row is an object itself from the Nature Communications article from Bill Poo's lab. And we have different column identifiers that we can look to help specify, for instance, you know, when I share the fully populated database, we'll be able to see different organisms, different library types, linking the geo and encode IDs, sample names, as well as what annotation model we use. So in this case, I annotated it using the bioconductor TXDB package. And we have other metrics that we're going to be integrating into this landing page. So, you know, just to float another idea. So you know, for instance, here we indicate the resource name is the Cardiovascular Development Consortium. This is the version 1 data set, I believe. But after we coordinate with Bill Poo's lab, maybe we can coordinate some uniform processing, have this as a version 1. I indicated myself as the data wrangler, and then Joe Yost, and I should probably also put Bill Poo's name here for the CDDRC wrangling into their Mosaic database. So this database platform is not just, you know, a meta summary of the data sets. We can also have some minimal analysis. So what I'm doing is I've launched a default configuration software panel of the data set. So this is looking at their embryonic stem cell, and I believe this one is for specifically looking at Vista neural enhancers within their library design. And you see here, each of these panels corresponds to some quote unquote slot within the data set. So for instance, row data is a slot that indicates all of the candidate regulatory elements that were profiled in this MPRA. So this is just a unique identifier for the gene ID and the name, since you have many-to-one mapping. But we have the gene symbol here and the gene ID, and this corresponds, so the row data information here corresponds to the assay matrix, which has the log ratios of our RNA and DNA. So that's what is in the assay, and the feature just means that this is this particular feature. So we have DNA average, and we can look at different, the DNA average or the RNA average. And right now, this isn't giving a lot of information, right? So it's just one sample point. But when we integrate all of these samples together, we'll be able to have like a violin plot showing all of this, the activity score ratios for all of these samples within some region of interest. So this is in active development. I just wanted to share that little teaser. And then you know, another reason why we're developing this database is that we do see that an enhancer loss of MYL2 and MYH7 does result in a phenotype that's consistent with heart disease in a mouse. So the Vista Heart Enhancer folks developed this catalog of using heart epigenome data, namely ChIP-seq, from whole genome sequencing studies, as well as mouse studies, to identify regions that they could functionally validate or explore. So in this case, they identified an enhancer for MYL2, and when they knocked this out, it had it had a phenotype that was of hypertrophic cardiomyopathy and dilated cardiomyopathy. So this is just, you know, as a another reason why we want to start developing these databases and standardizing the meta attributes so that we can systematically evaluate the loss of function for all of these enhancers given some consensus MPRA models. So, you know, we really want to start thinking about building an object-oriented database for MPRA data, because there's just so many varieties of reporter assays in terms of the experimental techniques. So Bill Poo's lab uses the adenovirus in an in vivo system. Our preliminary cardiac MPRA is going to be employed in lenti MPRA and in an in vitro WTC11 cell line. So there's, you know, different cell types and organisms and experimental techniques. So a lot of different open-ended utility. And the domain biology varies from congenital heart disease to cardiometabolic. And in terms of the choices in technology, there's different modes of integration: chromosomal, episomal. And so this database is really catered towards creating a central reporting and version control platform for sharing MPRA data and analysis. And so here we're just building on established object designs, namely a summarized experiment object, to support this systematic and integration of MPRA data sets. And yeah, so again, this is just what the IC MPRA base looks like. So IC stands for Interactive Summarized Experiment Explorer. And this is for sharing both the data and the code too. So for instance, what we're looking to do is create an open source GitHub community that people can help share, whether as a data wrangler or as somebody that's interested as an analyst for a secondary or tertiary analysis evaluating transcription factor binding motif. So lots of different open-ended utility just for the database and the reporting that we have ideas for. And so this is all leading up to a push-button solution for MPRA analysis. So we have an open source organization called MPRA Hub for our analysis and sharing in different genomics cloud computing infrastructures. The biodata catalyst is really what's driving a lot of this. So in the IGVF consortium, what we've proposed is to have a configuration file where users defined a list of NGS identifiers to launch the MPRA processing workflow. And so the idea here is that you're just going to drag and drop your files, press execute. And what this will do for the analyst is it'll pull from one of our repositories in our open source MPRA hub and take that pull request and auto-generate a quality control notebook that the analyst will review before submitting their object to our database, which is hosted on Synapse. And so they'll also be able to contribute to the IC MPRA base analysis platform, either their data set or their analysis. And we're showing how to do some of those workspace demos for creating those application plugins just for the IC MPRA base within Terra. And the goal here is that we'll be trying to semi-automate release of our MPRA genomics index catalog on Anvil each quarter. So this is a culmination of what we're trying to lead up to for our MPRA computational wheelhouse. So next steps, we are looking to release the MPRA Snakeflow processing application on the NHLBI biodata catalyst soon. We're stress testing that right now, and I think Dave will talk a little bit about also like different development strategies of how we've used SnakeMake. We'll also be publishing our open source MPRA database. It consists of roughly 130 data sets right now that have been formatted into a summarized experiment object. And then we'll also be defining what our primary and secondary minimal standard operating procedures are for analyzing MPRA data. So just quick acknowledgements. First, thank you to the AHI2 lab at UCSF for an amazing opportunity to learn more about functional genomics and design an MPRA, investigating MPRA, as well as Benoit Bruneau at the Gladstone Institutes for his mentorship in training me, and also for the opportunity to learn about the PCGC and bench to bassinet program. And UC San Diego to Neil Chi for sharing the CVDC version 1 data set, and just always being somebody that I feel like I can chat with and get real world advice on about just the scope and scalability of some of the ideas that we have, as well as IGVF and ENCODE for helping us organize and work with experts in the field to design the experiment and just statistical parameters for designing the MPRA, as well as the biodata catalyst and the bench to bassinet and CDDRC program, and all of the computational programs that help get these projects up and running. So so some of the experience that I've had as a B2B fellow and using the biodata catalyst is that, and just in terms of skill development, what I'm learning is that there's not one data sharing and analysis strategy that fits all, that each project has its own special nuances, either in how you analyze it or how you share the data. There's a lot of research and networking opportunities. Our office hours every Thursday that we attend. There's other fellows that I'm starting to meet from TopMed, as well as the PCGC and CDDRC program. There's access to resources that I am really excited to begin working with outside of MPRA. And I'm not an epidemiologist, but I'd love to learn how to identify common and rare variants and de novo variants, and just explore the VAT ecosystem within the BDC. It's just really, a really great exposure to the cutting edge research tools that the BDC is helping support, just in terms of the accessibility to young researchers that, in my opinion, are really going to help trainees find career opportunities after their PhD. So advice, ask questions, and don't be afraid to ask questions, and maybe start with an established data set or model that you can collaborate with the experts to understand and implement. And then, one of the things that I was really excited about learning at the beginning was like, how do I make one of these workflows? And just, taking it slow, being incremental about your development strategy. So start off with a tool before implementing a massive workflow, which you kind of have to do anyways. So, all right, I think I'm passing it to Dave now. Unless there are any questions, maybe I should wait to see if there are any questions for a couple minutes.
Andrew, can I ask a question?
Yeah.
Really nice work. This is Chani. I was wondering about kind of the compatibility of the different types of data sets in terms of cross species, like evolutionary conservation, because a lot of these crees are going to be identified in people, but obviously might be tested in animal models for pharmaceutical development.
Yeah. Yeah, I have not. So I haven't done like a fast cons analysis between the IP, like the in vitro human embryonic stem cells with the mouse in vivo and in vitro. That's a great question. That's something that definitely should should get done and reviewed before building on the in vitro assay for an in vivo application. Though what we did do for the first MPRA is we did do intersections with in vivo human and mouse from the Vista Heart Enhancer in the Cotney lab. But in terms of the evolutionary conservation and the activity of those sequences, I'm not sure what we would have to have that as a retrospective analysis, since
For
the data
sure.
has already been we're already starting to generate the data. But that's it. Yeah, it's a good question.
Awesome. Really nice work.
Yeah, thank you.
Any other questions for Andrew? Thank you so much, Andrew. That was a really great presentation and super exciting to the fact that you're bringing this sort of multi-omics type analysis to the BDC community. So.